Well, hello and welcome to everyone joining us for this Xtalks webinar. Today's talk is entitled Viral Vector Manufacturing Success Stories, Onboarding to GMP Production. My name is Corey Stanton and I'll be your Xtalks host for this event. Today's presentation runs for approximately 60 minutes and the webinar includes an interactive Q&A session with our speakers. To make the most of this session, feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to as many as possible. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. Please note that all participants are in listen-only mode and that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Oxford Biomedica, who developed the content for this presentation. Oxford Biomedica is a quality and innovation-led cell and gene therapy CDMO with a mission to enable its clients to deliver life-changing therapies to patients around the world. As one of the original pioneers in cell and gene therapy, Oxford Biomedica has more than 25 years of experience in viral vectors, the driving force behind the majority of gene therapies. Headquartered in Oxford in the United Kingdom and with additional locations in Oxfordshire, UK, Lyon and Strasbourg in France, and near Boston in the US, Oxford Biomedica collaborates with some of the world's most innovative pharmaceutical and biotechnology companies, offering world-class capabilities that span from early stage development all the way through to commercialization. One of our speakers today is Dr. Amy Barker. Dr. Barker serves as principal scientist in Oxford Biomedica's process development department. Her team manages the initial onboarding of new client products, conducting tailored feasibility studies that encompass screening multiple genomes, assessing plasmid ratios through design of experiment strategies, and evaluating platform performance using relevant analytical assays in both indicator and target primary cells. Prior to joining Oxford Biomedica, Dr. Barker gained extensive postdoctoral experience, specializing in applying her expertise in cell and molecular biology to enhance human health. Joining Dr. Barker today is Dr. Andre Raposo. Dr. Raposo directs Oxford Biomedica's innovation department, leading scientists in pioneering cell and gene therapy research by employing computer-aided biology and high-throughput automation. With extensive experience in technology development, upstream process, DOE strategies, analytics, robotics, and regulatory compliance, Dr. Raposo previously contributed to GSK's Transduction Sciences CGT platform, CMC. Earning his PhD from University of Oxford, he focused on host restriction factors controlling HIV replication, which he then explored further during postdocs at the University of California, San Francisco, and the George Washington University. And rounding out the panel today is Hannah Boss. Hannah is the head of quality control with responsibility for product quality testing, stability, contamination control, and analytical method lifecycle management. She has 16 years of industry experience, including roles in research, analytical development, and quality control. Prior to Oxford Biomedica, Hannah was the head of quality control molecular biology for Homology Medicines, Inc. Hannah holds a Bachelor of Science in Biology from the University of Massachusetts Amherst and a Master of Science in Biotechnology from Worcester State University. Now, without further ado, I'd like to hand the mic over to our very first speaker today, Dr. Andre Raposo. Andre, you may begin when ready.
Thank you very much for the introduction, Corey. Thank you very much to everyone. So I hope you can hear me and see me well. So I'm delighted to be part of this webinar today. And I'm joined by my colleagues, Amy and Hannah. And the title of our webinar today is Viral Vector Manufacturing Success Stories from Onboarding to GMP Production. So what will you learn today? So you'll learn about client success stories from early onboarding to GMP manufacturing of viral vectors for clinical use. You'll also learn about cutting-edge end-to-end automation of analytical assays and design of experiments, DOE, to optimize vector production. You'll also hear considerations for product comparability during process changes and strategies to enhance viral vector production. At the end, you will hear a success story from PD, process development, to GMP, involving cross-functional teams. So the agenda for today is as follows. I will be introducing the company, Oxford Biomedica, followed by the advanced analytics and DOE strategies. Then my colleague Amy will provide an update on the client product onboarding with a particular focus on a lentiviral vector case study. And then my colleague Hannah will discuss the process transfer from development to GMP, particularly on the AAV case study. And then we will have time for Q&A. So without further ado, I'd like to introduce to you Oxford Biomedica. So Oxford Biomedica is an innovation-led CDMO, contract development and manufacturing organization, specializing in adeno-associated viral, lentiviral, and adenoviral vectors. We are a leader in viral vectors, enabling the delivery of life-saving cell and gene therapies. We have over 25 years of proven experience offering complete development and manufacturing services of all vector types. We have established a global operation infrastructure with facilities here in the UK, facilities in the US, in Boston, Massachusetts, and more recently in France, in Strasbourg and Lyon. We have successfully to date manufactured over 380 GMP batches of multiple vectors, and we have presence in over 40 countries with approved products using our viral vectors. We have robust processes and a deep expertise in viral vectors. Our innovation engine has delivered multiple breakthrough technologies. I will talk particularly about lentiviral vector technologies. We provide cell and vector engineering, which is essential to increase capability and productivity. And recently we have launched our 4th generation lentiviral vectors with TetraVector, which is an Xtalk. So you will be able to download and see the talk at your pace. We also provide producer cell lines using our lenti-stable technology. We provide large-scale bioprocessing to increase yield and improve purity of our products. We offer perfusion systems with our enhancer technology, our U1 snRNA, and our small molecule I3A that improve and boost titers. We have invested significantly in digital transformation to maximize data integration and big data analysis. We offer design of experiments or DOE services powered by automated systems, which I will discuss in a little bit. We also offer extensive and exceptional expertise in analytical development, characterizing our vectors to achieve rapid batch release and QC testing. We offer cutting-edge technology and in-depth characterization of our vectors using proteomics, transcriptomics, and other advanced analytical tools. Now, I will discuss a little bit about our advanced analytics and DOE strategies. Now, Oxford Biomedica has developed and optimized the analytics required for process commercialization. We offer superior analytics to allow us to understand both the vector attributes and process performance through development and routine QC testing. However, there are challenges associated with the analytical methods that characterize complex structures such as lentiviruses or AAV vectors that require specific and specialist knowledge, techniques, and equipment. So therefore, we offer a comprehensive suite of in-house assays, ensuring the full vector characterization, quality control, stability testing, and preparing CMC modules for regulatory filings. We also offer custom-made assays for specific applications or types of products that our collaborators demand. So in terms of automation, Oxford Biomedica has invested significantly in this infrastructure to support analytical development. So we offer automated qPCR workflows that are ready to start with full traceability in research and GMP compliance laboratories. We also offer automated tissue culture workflows. We are proud to be one of the few companies in the world that can offer in-house GMP compliant RCL co-culture assays. And we also offer automated cell culture transductions for vector titer determination. And we have recently made a case study with Hamilton Robotics that I would invite you to search for us and read about it. We also offer automated ELISA setups, automated mass spec for sample preparation, and importantly, data integration, analytics, and visualization through design of experiments. So all in all, this allows us to have faster, more reliable, and more robust analytic portfolio. Now, at this stage, I would like to introduce a team that I'm very proud of, which is the computer-aided biology. So our computer-aided biology group is made by leading scientists and a very interdisciplinary team from background in biostatistics, immunology, virology, and engineering. And computer-aided biology combines tools that augment the human capabilities in biological research. It's made by the digital, which is powered by AI, includes the software designed to simulate biological systems, as well as collating, connecting, and structuring, as well as analyzing experimental data from wet lab experiments. Now, this is enabled by automation in the physical, which includes systems that allow the seamless transfer of simulated biological designs into real wet lab experiments through abstract experimental design, simulation, and execution. The DOE sits in this, which is a service that we provide, particularly for optimization of transfection and transduction workflows for any platform. So for those of you who are not familiar with DOE strategies, DOE, or design of experiments, is defined as a branch of applied statistics that deals with the planning, conducting, analyzing, and interpreting controlled tests to evaluate factors in a given parameter. So it allows multiple factors to be manipulated, determine their effects in a specific output or response, all at the same time, which enables then the identification of important interactions that might have been missed when experiments are performed as a one factor at a time. So all possible combinations can be investigated as full factorial, only a portion of it as fractional factorial. So we have been employing this to optimize our upstream lentiviral vector titers, which is essential for the development and manufacture of commercially viable technologies and cell and gene therapy products. So just to give you an overview of how this works, typically in a classical one factor at a time experiment, one would be investigating different concentrations of a particular variable, in this case, genome. And this would be manipulated in a manual, relatively small scale experiments, which are obviously prone to error and relatively variable. So what DOE allows you to do in a multifactorial experiment is to combine all of these parameters all at once and understand how the interactions between all of these parameters play with each other, and therefore optimize your system to a specific target, either titer or transduction efficiency in primary cells or your cell of choice. This is fully automated, allowing hundreds of experimental conditions done at any given time. How does this look like in silico? So we have developed the design, either determined by computer, or a client or partner would be able to select their particular design of choice. Then the design is sent to a liquid handler, a robot, and then all the analytics are carried out and the best model is fit to that particular parameter. If there is a need for additional iterations, we can do this using our curated database that provides additional information for the variable. Now, just to give you an idea of how this looks like in real, in a real experiment, here you can see different parameters given at any given time: your genome, the different packaging components, your enhancers and boosters, scale, and different genomes as well. So what you see here is a combination of all of these factors together at different concentrations, which then allows you to plot a 3D interactive graph to see where the space of that particular experiment is best suited for the output that you want. So with that, we offer exceptional knowledge, technology, and expertise. We have over 25 years of proven experience in manufacturing vectors. We offer flexible solutions with leading platforms that permit rapid scale-up. Our cutting-edge technology, driven by digital transformation, is enabling full vector characterization, quality control, and testing. And being an innovation-led CDMO, innovation is at our core, and it has delivered multiple breakthrough viral vector technologies. And that enables us to be a leader in viral vectors, delivering life-saving gene and cell therapies. And with that, I would pass over to my colleague, Amy.
Thank you, Andre. Let me just share my screen. So I am part of the client onboarding team here at Oxford Biomedica. And as part of that, we carry out a lot of projects to onboard new clients and to carry out small-scale assessment to select the optimal conditions for scale-up for each project. Now, my work focuses mainly on lentiviral vectors. So while this section will largely focus on lentivirus, the processes I'm going to talk about will apply to all the different vector types. Here you can see an overview of our client onboarding process. And what happens at the beginning is that we come in at this blue section here, and the first thing we will do is we'll discuss with you, the client, what DNA constructs are required to carry out the work for your project. Now, we can onboard an existing genome you've worked with before, or we could clone your transgene and your promoter of choice into one of our standard genome backbones for use with our platform. We then arrange for procurement of the DNA to carry out the initial work. And once we have that DNA in-house, we proceed with the initial assessment. Now, that's normally carried out at small scale in shake flasks. And the initial testing is first and foremost to ensure that your vector transgene is going to work well with our platform, and also to give you an idea of the expected titers within the project. However, we offer a range of optimization options which can cover multiple genome constructs or multiple plasmid ratios. After this stage, we analyze the vector we've produced and we discuss with you the best options to proceed to scale up. And the project then proceeds to the 7-litre and then to the 50-litre bioreactor scale. In terms of genome production, we offer a range of different backbone options for our lentivectors, including our modular tetravector backbones, which Andre mentioned. There is a webinar available on Xtalks about those as well. We clone your transgene and your promoter into one of these backbones, and then we arrange for small-scale DNA production sufficient to cover the initial studies. And we can also then begin to arrange GMP-grade plasmid production for later on in the project. With the plasmid, we will perform nanopore sequencing to ensure we have at least 4-fold coverage of the whole plasmid and not just of the cloning junctions. We're happy to arrange for cloning and testing of multiple constructs. So, for example, if you wanted to test a variety of different promoters or you had different transgene variants, we can make all of these and assess them in parallel. Alternatively, we can onboard your own vector genome, at which point we can proceed straight to small-scale vector assessment. In terms of vector genome production, for our initial shake flask studies, we offer a standard genome plasmid evaluation where we can look at one genome or multiple genomes using 3 levels of genome plasmid. We would use standard platform ratios of helpers and transfection reagent for this. Alternatively, we can offer an in-depth bespoke DOE-guided plasmid ratio assessment where we may vary the quantities of plasmids, including the genome and the helper plasmids, or we may change things like the concentration of transfection reagent, and this will go through the automated workflow. At this stage in the project, we would also look at evaluating our lentivector titer enhancing technologies, so the I3A small molecule and the U1 snRNA plasmid, to see if they would be beneficial to your vector. And once we've produced the vector, we would analyze this with various different outputs. So we can look at transgene expression via flow cytometry. We can look at functional titer, so integration of vector in the case of lentivector, and we can look at particle quantitation. We can also ship these samples to yourselves for analysis in your own systems while this is going on. Now, one question you may have is, why would I want to do a plasmid ratio assessment? Now, our platform is excellent and it works very well for the majority of transgenes. However, there are some cases where a full assessment may be optimal for your vector. In particular, if you have a large transgene, you may find that you need to adjust plasmid ratios to get optimal vector production. Or alternatively, if your vector produces a little bit of cell toxicity, you may benefit from using our trip repression system, which suppresses transgene production during vector production process. If you want to use your own genome backbone or your own helper plasmids, that's also okay, but it may not work optimally with the plasmid ratios that we use for our own platform. So in that case, you would also want to do a plasmid ratio assessment. And lastly, our platform is optimized for titer on HEC293T indicator cells. However, you may wish to use a more clinically relevant target cell. We do offer T cell assays for products such as CARs, which are used in T cells, and we can also onboard other primary cell assays if you require. Now I'm going to talk a little bit about a case study from one of our clients, and this client came to us with a T cell directed CAR product, and they asked for a full vector plasmid ratio assessment. So to properly pinpoint the best conditions for them, we designed a custom DOE for this client. This was based on a central composite design, but it was optimized a little bit to have a bit more statistical power around the edges of the design space, so at the far ends, and a little bit less repetition towards the center of the design space. And you can see that design space in 3D with the graph there. Each of the 4 factors that were selected, this was genome, GAG-POL, the VSVG envelope, and the concentration of transfection reagent, were varied over either 4 or 5 levels. And those levels were selected partly from discussion with the client to see what had worked for them before, and partially from historical data within our platform for similar transgenes. We also assessed the impact of our U1 and our I3A technologies, and that meant that by the end of this design, we had 24 conditions, each of which we assessed in duplicate. To ensure that our small-scale production is as similar as possible to the scaled-up bioreactor, the cells that we used for the vector production were obtained from the bioreactor on the day of transfection. Now here you can see an overview of the workflow. We produced the vector in the range of conditions at the small scale in shaped flasks, this vector, and then we clarified using a standard filtration. As well as the clarification, we also carried out a column purification to make purified vector samples, and both the clarified and the purified vector were tested on our HEC293T indicator cells, and we assessed the vector success in these cells by transgene expression using flow cytometry and vector integration using qPCR. We also tested the purified vector samples on primary T cells from multiple donors, and in this case we didn't test the clarified samples because impurities at this small scale stage can cause some toxicity in primary cells and confound the results. The T cells were also analysed for transgene expression and for vector integration. When we looked at these results in the HEC293T cells, we saw that several conditions surpassed our standard platform for this particular vector, and you can see that our standard platform here is the orange line. We do have some conditions that are far below this, which is what you'd expect with a DOE exploring the design space, but quite a few that are above. Condition 21 was the best. However, 4 or 5 conditions had very high performance in this assay. When we examined the same samples on primary T cells, we also saw several conditions that outperformed our platform. And again, you can see the platform here with the orange line. The overall pattern was similar, so high performers in the HEC293T assay also performed well in the T cell assay, although the changes are much more pronounced in the T cells. The pattern, although it was similar, was not exactly the same. So the best condition here was condition 4, while in the HEC293T cells, it was condition 21. We then input both sets of data into our DOE modelling, and we analysed them to identify the best production conditions, both for the HEC293T cells and for the T cells. To the left, you can see the 3D model data for the 2 components, which is genome and GAG-POL in this case. And you can see that this produces a clear parabolic curve for each. So the advantage of this is that we've properly captured the design space with the best titers in the center of the design space and the lowest titers at the edges of the design space, which means that we've definitely captured the best levels here. On the right, you can see the desirability curves for the HEC model for each of the 4 components that we examined. And we've mapped condition 21, which gave us the best HEC titer, onto these graphs with the blue dots here. And you can see it sits quite close to the optimal level for each of these components, which explains why it performs so well in our HEC293T assay. Following this, we had a discussion with the client, and while the analytics were being performed at Oxford Biomedica, we'd also shipped samples of this vector to the client, and they'd performed their own T-cell assays at their own facility. And they'd seen a similar pattern of best performing conditions to us, which was great because it added a lot of additional weight to our analysis. We discussed various recommendations for scale up, and in this case, we provided multiple possible conditions that could be taken forward. We outlined the benefits of each, taking into account things like how close they were to the optima for both the HEC and the T-cell assays, how feasible the scale up was in terms of certain quantities of DNA, and also potential batch costs in terms of different plasmid quantities. The client selected the condition which fit their requirements best, and this progressed smoothly to the 7-litre scale, where we produced 2E10 TU of vector following full downstream purification and concentration. That was sufficient for the client to perform the studies that they needed to reach their next milestone, and they then progressed to our GMP manufacturing team, where this vector is now being made for treatment of patients. So I will now hand over to Hannah, who's going to talk to you a little bit more about the transition to GMP for projects in an AAV setting.
Thank you, Amy. So now it is my pleasure to share a case study from our Oxford Biomedica site here in the US. During this project, we successfully transferred a new process from our PD group into our GMP facility on behalf of our client. To start, I'd like to go over what the project goals were. For this particular client, the goal was to make fewer batches that would provide higher yields per batch without sacrificing product quality. This particular client we had a strong partnership with, and we had ultimately manufactured 46 lots for them successfully at our AAV facility here in Bedford. In order to implement a new process, we had to transfer process 2 into our GMP facility. And we also had to do this during an ongoing phase 1 study for their lead candidate. Some things that we implemented here at OXV in the US were things that would help the client amend their IND for the ongoing clinical study. We provided a strong comparability package. We also coordinated our CMC timelines such that we could utilize this innovative process too during an ongoing clinical study. So how were we able to increase our process yields? What I would like to share now is a timeline of how our AAV platform process here in the US has increasingly changed over time. With process 1, what I'm showing you here is we were able to generate at the 500-liter bioreactor scale about 250 milliliters of drug substance at 6E13 vector genomes per mil. However, with process 2, we were now able to implement process changes that resulted in the equivalent of the same 500-liter scale, now generating 2.5 liters of drug substance at the same concentration of 6E13 Vg per mils. We used some key aspects of our upstream process optimization to achieve this. Things like using a dual plasmid system, optimizing our bioreactor conditions, utilizing a more productive cell line, and ultimately implementing a novel transfection system such that we could achieve where we are today. And this is something that we wanted to deliver for this client. Now, when we talk about higher yields, that implements the ability to treat more patients. So when you look at our ability to treat patients with our process 1, each manufactured batch could ultimately be used to treat 3 patients. Now, when we utilize our process 2 system, we are now able to greatly increase our ability to treat more patients. Now, with that same process and that same product, using process 2, we can now treat 34 patients. And we can also greatly reduce our cost per patient. Ultimately, the project team and the client agreed this was the best way for us to deliver our product to more patients who are in need. Now, we had a high success rate in our GMP facility with manufacturing process 1. We had already made 50 GMP runs at our 500 liter scale, and we had a 98% success rate. So ultimately, we never had a delay in our clinical supply for our clients, and we always had drug product on hand via process 1. However, we had never made process 2 in our GMP facility. So what we did as an organization is we made it clear we were going to provide the same level of execution and quality to this new process, and we would also have to show comparability of the product along the way. Now, our comparability strategy really highlighted our experience with process 1. We relied on all of the data we had generated via process 1 to determine what we thought acceptance criteria would look like. And what we've done is we've determined, using our average of process 1 results, acceptance criteria that we could use to compare what we were getting with our process 1 to the lots that we were generating being process 2. The range we determined would be 3 standard deviations away from that average. And if our results from process 2, we were able to ultimately utilize a comparability strategy that showed we would be able to move forward with process 2. So how do we tech transfer here at our Oxford Biomedica site? We have a highly efficient process. We are streamlining our tech transfer process such that along the way we're focused on efficiency. We would like to always give deliverables to our clients that would include things like risk assessments, product batch records, test methods, as well as a solid comparability report at the end of the project. This is really meant to bolster the client's overall package. One strategy that we utilized, which I wanted to highlight here, was how we were able to use our engineering run material to essentially impart a strategy where we could utilize that engineering material as representative material to perform method qualifications. We use this to determine our acceptance ranges, and we also use this to determine that our GMP1 manufacturer was acceptable for release testing. I'm now excited to share our GMP data, where we were comparing our material generated from process 2 to our process 1, and I'm going to stop here so that I can show you how we were able to compare 2 key product comparability things. The first one would be our percent of empty capsids that were generated. We wanted to compare what we achieved from process 1 to our process 2 batches. As you can see, we have comparable percent empty capsids generated, if not improved, via process 1. The other key attribute that I wanted to highlight was our relative gene expression assay. This is where we're really looking at our potency of the final product when compared to process 1. In all cases, our relative potency was comparable to the reference standard, and our process 2 GMP1 obtained a relative potency value of 92% when compared to our process 1 and our process 2 reference standard. Overall, what this is showing is we have comparable potency and comparable full capsid ratios in our final drug substance. I also wanted to highlight our comparability of our process residuals. This is a good indication of product quality over time. The key component of quality here is that we were seeing comparable residual profiles between our process 1, our process 2 engineering run, and our process 2 GMP 1. We overall saw similar trends and found that using this acceptance criteria we had set to achieve in the beginning, we're seeing comparable levels of impurities between our process 1, our process 2 engineering 1, and our process 2 GMP 1 lot that was manufactured. Ultimately, we were able to generate a lot that was dispositioned for clinical use. We were able to generate 95 vials of drug product at 10 mils per vial and provide that to the client for clinical use. We dispositioned drug substance and drug product lots for this particular campaign on time and within their specifications. We were able to use this as a way to add it to our list of right first time lots. Now, what we consider right first time is that we don't have any specifications that were outside of what was exceeded. All of our testing was within the specifications outlined, and we had a limited number of deviations that showed the process and the release testing was executed properly. So we were really excited with this result, and we were able to deliver this on time for our client. So in summary, we hope that this case study would highlight what our team can do. We are really committed to helping our clients' programs succeed. Whether your goals are to tech transfer your process into our facility, utilizing our highly efficient process 2, or making process changes, we are here to work with you to enable your late stage achievements and help you with your clinical supply. So the key takeaway here that I wanted to highlight, by making all of these process changes, we greatly increased our product yield and ultimately were able to treat more patients with a similar product profile with process 2. And with that, I will turn it over to Corey for the Q&A. Thank you.
Excellent. Thank you very much for that insightful presentation, everyone. We are now ready to begin the Q&A portion of the event. And as we do so, I invite you to continue sending questions and comments using the questions window chat box. It does look like we've already received some audience questions, so I will go ahead and start with the first. This question is for Dr. Andre Raposo, and it's actually a two-part question, Andre. So first question here is, are you offering commercial production? And the second part of the question is, can you share what scale or volume you run your DOE for upstream parameter optimization?
Thank you for those two questions. So yes, we do offer commercial supply. We have some customers that have approved drugs in the market, so it is commercial supply. For the second part of the question, it's an interesting question. So we do offer design of experiments at multiple scales. So typically, experiments can be initiated in a research environment, in the innovation department, at small scales, such as Ember 15, for example, shake flask scale, or macro titer plates as well. Things then scale up to bioreactors, 5 liters, 7 liters, Ember 250s, and then eventually larger scale, depending on what the customer will require. Yeah.
Thank you for that answer. The next question here from an audience member, I believe this one is probably suited for Dr. Barker. The question asks, is the automated plasmid mix ratio assessment then functionally tested on cells, and what is the readout of the plasmid ratio assessments?
So we test all of our vectors with a functional test on cells. So all of the vector that we produce during the plasmid ratio assessment is tested on at least the HEC293T indicator cells, and we can read out for transgene expression using something like flow cytometry with an antibody, or we can test for vector integration into the host genome to get a vector copy number. Or we can look at primary cells and do the same readouts on those cells. So everything is always tested with a functional readout as well as any particle quantification.
Excellent. Thank you very much for that answer, Dr. Barker. I have another question for you, actually, from an audience member here. They say that they believe you're using a third-generation lentiviral system, but in your DOE study, they mentioned that they didn't see REV on the list. So they're just wondering whether you included it.
So we are, for that particular study, was a third-generation lentiviral system. We didn't include REV in that particular case study, as the client hadn't previously seen much effect with it, and so they chose to focus on other factors. But this is one of those things where these studies are generally quite bespoke, and we can look at as many or as few factors as needed.
Okay, great, thank you. Keeping with you, Dr. Barker, I've got another question for you here. This audience member asks, how long does it usually take from coming in as a new client or client onboarding to then reach GMP production?
So at the very fastest you can go, where everything works very smoothly through process development. It usually takes a little over 6 months, but that will depend on how much process development is needed before GMP.
Great, thank you. Moving back to Dr. Raposo here, we have a question for you. What has been the biggest improvement by the introduction of automation into your analytical workflows?
Yeah, that's a good question. So we have been introducing automation at Oxford Biomedica for the past 6-7 years. And over this period of time, what we have noticed is that where we have improved has been mainly in precision. So our assays, as you can imagine, need to be done in multiple replicates. So we managed to decrease that standard deviation that assays can have when you're performing them. So that has been a big improvement. Obviously, by the introduction of automation, you're able to have assays which are more robust, and perhaps the main improvement has been operator time. So while the robots are doing the job, you can go about and do other things that scientists do, analyze data, plan experiments. So it has been a change in how operators perform their experiments. You can perform experiments and leave them overnight. The systems will not sleep, so they will be able to perform all of these operations at different times that are not necessarily the times that people are there. So and obviously as well, personal development, people have other tools to develop their skills, and learn coding, for example, has been a big positive as well. So yeah, that's sort of it.
Great, yeah, thank you very much, Andre, for that. This next question here is for Hannah, actually. This audience member is asking, what are the key operational aspects that result in a right first-time manufacture of drug product?
That's a great question. So I think in order to be right first time in a GMP facility, you have to put the right people, tools, and controls in place to maintain a successful operation. So some things that are important: having people that are well trained with the right expertise, having the right mindset around quality is important, and making sure that our systems are efficient and they're right for their intended purpose. Some other things that we do is we put a lot of controls in place because failures do inevitably occur. And we want to make sure that even if we do have failures, we prevent them through these additional controls. So I would say by prioritizing these different areas, and we're always continuously seeking to improve upon them, we really can make sure that the execution is going to lean towards right first time.
Excellent, great, thank you. Moving back to Dr. Barker, we have another question here that's asking, have you already produced lentivirus batches with another pseudotyping, basically other than the VSVG?
Yes, we have produced vectors with various different envelopes for lentivirus. I'm not sure how far, I don't think any of our commercial products currently have non-VSVG envelopes, but we definitely have some of these in development.
Great, excellent, thank you. Sticking with you here, Dr. Barker, this audience member is asking, do you find that the DOE modeling for the HEK293T and the primary cells usually agrees? And if it doesn't agree, what do you end up doing in that situation?
So this actually happened in the case study that I presented to you. The HEC293T and the T-cell assays produce slightly different models, which was one of the reasons that we recommended various different conditions that could be taken forward to that client. In that case, we will usually discuss the various different options with the client. We will explain the pluses and minuses of each one, and the client can either choose to go forward with one of the conditions we've already done, or we can go back and do another DOE that explores a compromise space between the two models to really pinpoint what that ideal ought to be. And in that case, it could be weighted in the way that the client wants. So if, for example, they want 75% on the primary cell assay and 25% weighting to the HEC293T assay, we can do that kind of thing.
Excellent, thank you. Switching gears back to the automation side of things, Dr. Raposo, I believe this question would be best suited for you. What do you find the role is of AI in the actual manufacturing process, if any?
That is a terrific question. So obviously we are an innovation-led CDMO, so we are always looking forward to new technologies and new advancements in the field. Obviously with AI and during the manufacturing process, because it's highly regulated, it's quite difficult to implement AI, not only for proprietary reasons, but you know, with everything that is related to AI. We have been experimenting with AI, particularly in the research department, with the data that is obviously not client sensitive. And we have been experimenting with the analysis of data and how the data can be sort of portrayed in a way that is easy to understand. At the same time, AI can be used to detect patterns in the data that we may not necessarily be able to detect with our normal human eye. So we are experimenting with AI, but in terms of manufacturing and regulatory compliance, we're not there yet.
Excellent, thank you, Dr. Raposo. Switching back to Hannah now, this audience member wanted to know, how can you best utilize the material generated during an engineering run for the late stage analytical strategy?
So engineering runs are great when it comes to utilizing it for your late stage strategy. They're great because you can use them for method qualifications, and ultimately a big part of the GMP success is having robust analytical methods. So when you perform an engineering run prior to your GMP manufacture, you can use the material generated from that engineering batch to complete method qualifications and also get an understanding prior to that first GMP release if you're having any issues with the analytics. The other thing that kind of helps with that, it helps with the overall C strategy. So we use the engineering run in a useful way to kind of provide a robust package of material that the client can then use for their IND enabling activities. So overall, using an engineering run, it's really going to strengthen your late stage regulatory filing. It gives sponsors more at-scale production runs, which ultimately gives the regulatory agencies more confidence in approving their late-stage changes.
That's great. Thank you very much for that answer, Hannah. Dr. Raposo, back to you. Interesting question here. What innovative technologies do you currently have on your pipeline that you're able to share, or is it all top secret? Can you tell us anything?
It's very top secret, and no, I'm able to share obviously a few. So as I enumerated in the slide, so our tetravector, I think, is our utmost technology that we are eager to implement. So this is the next generation, the fourth generation lentiviral vectors. So I would definitely invite our viewers to go into the Xtalk and have more detail on that. But also the lenti-stable and the TRIP technology that Amy mentioned as well. These are all technologies that are available to support the different client programs. Obviously, we have the perfusion system. We have the enhancers, the boosters, which will make a significant difference, in particular in products which by nature have lower titers. So these are able to bring them to commercially viable products, such as the U1 snRNA and our small molecule enhancers. But obviously, we're always developing new tools and technologies for specific cases that come to us. So this involves in analytics, cell line development, and also in process development. So it's a question of coming to us and discuss with us what you desire, and I'm sure we will be able to find a solution.
So not very top secret then, great. Sticking with you actually, Dr. Raposo, just for a moment here. You did mention you have mass spectrometry capabilities. Can you elaborate a little bit on where you're applying the proteomics in your pipeline?
Absolutely, with pleasure. So we do have a proteomics team at Oxford Biomedica in the UK. We have 3 top mass spectrometers that are top of the range from within the field. So what we offer routinely will be proteomics analysis that are not necessarily available in the market. So for example, certain types of ELISAs, you don't have a particular antibody. So mass spectrometry can properly quantify your specific analyte in a solution very accurately and very quickly. We're also providing deep characterization of host cell proteins and your lentiviral vector proteins within your product. Particularly, proteomics has really been fundamental for process development and characterizing every step of the way during upstream and downstream to get to the best quality product that we can offer and best performance. So yes, please come and talk to us if you're interested in proteomic services, and we'll be able to help you.
Okay, excellent, fantastic. We have time now for one last question here from the audience, and again, it's for you, Dr. Raposo. This person's asking if the creation of the computer-aided biology group, it's quite innovative. Could you just elaborate a little bit more on the group's remit within Oxford Biomedica?
Oh, certainly, with pleasure. I am very proud of the group. So we've been creating this group for the past 5 to 6 years, and we offer multiple disciplinary knowledge, as I mentioned, from immunology, virology, process engineering. So the group is created to support all the other teams within the company. So from process development, analytics, manufacturing, and early innovation. So the remit of the group is to provide automation, design of experiments, and basically solutions that you may not necessarily have as off the shelf, so development strategies for those type of technologies. So yes, it's a relatively new group, but it's contributing fundamentally for our platform.
Yeah, well, excellent. Thank you, all 3 of you, for your answers and for fielding all those questions. I know our audience really appreciates it, but at this point, we have reached the end of our Q&A session. Now, if we couldn't attend to any of your questions, or if you have any further questions, you can get in touch with the team at Oxford Biomedica using the contact info that's currently up on your screen, and they will have a chance to follow up with you after the webinar. Thank you once again to everyone for attending and for participating today. I'm currently adding a link in the chat box right at the moment where you'll be able to view a recording of this event. And you can share that link with your colleagues so that they may be able to register and view it as well. You will also be receiving a follow-up email from Xtalks that will provide access to today's recordings. And as we wrap up things here, please note that a survey window will appear on your screen, and your participation in the survey is always appreciated as it helps us improve our webinars. Now, please join me once again in thanking our speakers for their excellent presentation today. We hope you found this webinar informative, and we wish you a great rest of your day. Thank you, everyone.
Bye.